Nutrition, I welcome you all for this one-day national webinar on PCR principle, types, and application. We now begin the webinar with the prayer song by Shamala from FNBC second year. Over to you, Shamala.
गजाननं भूतगणाधिसेवितं कपित्थजम्बो फलसारभक्षणम् उमासुतं शोकविनाशकारणम् नमामि नमामि विघ्नेश्वर पादपङ्कजम् मातृदेवो भव पितृदेवो भव आचार्यदेवो भव अतिथिदेवो भव गुरुर्ब्रह्मा गुरुर्विष्णुः गुरुर्देवो महेश्वरः गुरुर्साक्षात् परब्रह्म तस्मै श्री गुरवे नमः. Thank you.
Thank you, Shamala. That sounded amazing. Now, I invite our respected principal ma'am, Dr. G. Achyuta Devi ma'am, to say a few words about our college.
Thank you. Good morning, everyone. I welcome all participants to webinar on polymerase chain reaction, principle, types, and applications. I'd like to extend a special welcome to today's guest speaker, Dr. Surya Prakash G. Ponnam, Assistant Professor, Department of Molecular Biology and Biotechnology, Tejpur Central University, Assam. Let me speak a few words about our college. Raja Bahadur Venkatrama Reddy Women's College was established in 1954 by Hyderabad Mahila Sangam, a non-profit educational society with a vision to empower women, especially those from rural areas. The college is the second oldest women's college in the Hyderabad. The college owes its existence to the foresight of Sri Raja Bahadur Venkatrama Reddy Garu, Kotwal of erstwhile Hyderabad state, Sri Burgula Ramakrishna Rao Garu, the first Chief Minister of erstwhile Hyderabad state, Sri Madapati Hanumantrao Garu, former Mayor of Hyderabad and Chairman of Legislative Council of Andhra Pradesh, Sri K.V. Rangareddy Garu, former Deputy Chief Minister of Andhra Pradesh, who were the pioneers in propagating women education in the state. The college had the rare privilege of welcoming Pandit Jawaharlal Nehru, the first Prime Minister of India, who laid the foundation stone. Dr. Sarvepalli Radhakrishnan, former President of India, who inaugurated the college building, and Srimati Indira Gandhi, former Prime Minister, who inaugurated the hostel building. The visit of Dr. APJ Abdul Kalam, former President of India, to address the students on the eve of Golden Jubilee celebrations is yet another landmark in the history of the college. Dr. C.H. Vidyasagar Rao Garu, Governor of Maharashtra, visited our college on occasion of Diamond Jubilee celebrations. Dr. Tamilsai Soundararajan attended the college in the eve of convocation. The college is recognized by UGC and affiliated to Osmania University, conferred autonomous status by UGC since 1989, selected by UGC as a college with potential for excellence since 2010, accredited with A grade by National Assessment and Accreditation Council from 2007 to 2017 and was awarded B++ grade in 2018 in the changed accreditation procedure of NAC. From humble beginnings, our college grown to mammoth size with over 2,900 students, providing hostel accommodation to more than 400 girls who hail from poor, the remote areas of Telangana. It is the first college to introduce BSc with Forensic Science course, MSc Data Science program. Presently, the college is offering 17 programs at UG level and 5 programs at PG level. And presently, We introduced BSc with data science and BCom with business analytics during the academic year 2020-21. PCR, that is polymerase chain reaction. This is widely used rapidly to make millions to billions of copies of a specific DNA sequence, allowing to take a very small sample a sample of DNA and amplify it. It is invented by Carrie Bank in 1983 and got Nobel Prize in 1993. Reverse transcription, that is a polymerase chain reaction, is a technique in which reverse transcription of the RNA into DNA takes place, and amplification of the specific DNA targets using polymerase chain reaction, which is used to detect COVID-19. In this present pandemic scenario, it is very important to know about what is polymerase chain reaction and what are the types, etc. I hope it will benefit all the students and faculty. Thank you.
Thank you so much, ma'am. Now I invite our respected head of the department, Dr. A. Rajni ma'am, to introduce about the department and few lines about the webinar. Over to you, Rajni ma'am.
Thank you, Renu. I am audible, ma'am?
Yes, ma'am.
Good morning, one and all. I welcome you all for a one-day webinar on polymerase chain reaction, principle, types, and application. It's my pleasure to welcome Dr. G. Ponnam Suryaprakash, Assistant Professor, Department of Molecular Biology and Biotechnology, Tezpur Central University, Assam. I also invite Professor Mutyam Reddy Sir, Secretary, HMVS. Professor Sudarshan Reddy Sir, RBVR Secretary, RBVRR Women's College. Dr. J. Achyuta Devi, Principal of the College. Dr. Kavita, Vice Principal of the College. I also invite my dear colleagues and student friends. Once again, I welcome one and all. Now I would like to enlighten about our department. A department is established in 1954, and we have introduced food and nutrition as one of the optional subjects at UG level in 2014. Department is supported by 7 faculty members, of which 4 are PhD holders and 1 are pursuing PhD. 2 are qualified NET. Department has 4 labs and well-maintained museum with rare collection of herbarium. Department has credit of having tissue culture lab. We have 4 MOUs, and we also have Eco Club, in which we conduct various student activities. Department regularly conducts seminars, workshops, extension lectures, and field trips to upgrade student knowledge. We also take up student project at UG level. The main aim today's webinar is to polymerase chain reaction, principles, types, and application. The present webinar may highlight on the principles and the techniques of polymerase chain reaction of identifying large copies of foreign DNA. The main objective of this webinar is to highlight RT-PCR techniques of the identifying virus DNA causing COVID-19 diseases. I hope all the participants will be immensely benefited with the valuable inputs shared by speaker of today. Thank you, sir. I hand over the session to Renu.
Thank you, Rajni ma'am. Now, I invite Dr. A. Kavita ma'am, Assistant Professor, Department of Botany and Food and Nutrition, to introduce today's keynote speaker. Over to you, Kavita ma'am.
Thank you, Renu. Am I audible, ma'am?
Yes, ma'am.
Yeah. Good morning to one and all. I, Dr. A. Kavita, takes the privilege to introduce today's guest speaker, Dr. Suryaprakash G. Ponnam, Assistant Professor, Department of Molecular Biology and Tezpur Central University, Napam, Assam. Sir has around 10 years of teaching experience as Assistant Professor, specialization in human molecular genetics and synthetic biology. To go with Sir educational qualifications, Sir worked as DPT Overseas Associate with Professor Nuts Tiger, head of the research group, Retinal Gene Therapy Laboratory, Department of Ophthalmology, UKGM Justin Liebig University, Gießen, Germany, from November 2016 to October 2017. Sir has completed his Doctor of Philosophy in Human Molecular Genetics from L.V. Prasad Eye Institute and awarded the degree from University of Hyderabad 2011. Sir did his Masters in Science in Biochemistry from University of Hyderabad, Telangana 2004. Sir completed his graduation BSc in Genetics, Zoology and Chemistry from Nizam College, Usmania University 2002. To go with the Sir research work, Sir has Sir has to his credit of ongoing 2 research projects, which are namely studies on genetic and epigenetics alterations on keratoconus patients from Assam, funded by DBT with worth of around 15.98 lakhs. Second research ongoing project, that is in vitro studies to investigate the therapeutic approach of editing the defective exon 3 of human CHST6 gene associated with the MCD through CRISPR technology, which is funded by DST worth of 45.94 lakhs. And Sir completed the project titled Diagnosis and Immunopathogenesis of Dengue virus infections, a longitudinal investigation into genotype diversity, diagnosis and immune response to dengue infections in Assam, Northeast India, which is funded by LSRB, DRDO, Government of India, worth of 65.10 lakhs. And when you go for the Sir's research publications, Sir has to his credit of around 12 research publications with around 156 citations. Not only this, Sir received 6 awards in his research experience of DBT Overseas Associate in the year 2014-15, JRF at national level ICMR in 2004, JRF national level CSIR 2005, and he has received Best Scientific Poster Award at Indian Eye Research Group held at Madurai, Tamil Nadu 2008. Sir also received Earlier Career Research Award in the year 2018-19. Sir has guided, under his guidance, Sir has around 4 research scholars at PhD level, 1 project assistant student currently working, and also 1 MSc student. Sir has conducted and also attended almost 35 seminars, workshops, refresher courses, and also delivered talks in national and international conferences and webinars. Sir, with this brief introduction, I welcome you once again, sir, for this one-day national webinar. Thank you very much.
Thank you, ma'am. Now I welcome you, sir, to take over the session.
Well, good morning, everybody. A warm namaskara to all of you. First and foremost, I would like to thank Dr. Achyuta Devi Garu, the principal of the college, for making this conference, live. And secondly, I would like to thank Dr. A. Rajani Garu, Dr. A. Kavita Garu, Dr. A. Rajani Garu for inviting me to this lecture, and Dr. A. Kavita Garu for a very, very vivid introduction of me. I think this is probably the best ever introduction somebody has given to me, and let me humbly accept that there are a few things which she has probably told out of colleague, respect for a colleague, a little more than what I actually deserve, to be honest with you.
Thank you very much.
And And I would like to thank Renu Arutla for this wonderful organizing, for the welcome speech or whatever thing she has done and she has been doing, and Shamala for her wonderful rendition of Vinayaka Stotram. For her, this thing. I would also like to thank Professor Sudarshan Reddy Garu and Mutyam Reddy Garu, who, I guess they are busy with some other navigations. So, but still, I wish to convey my greetings to them. I really am fortunate to begin this lecture with the talk of Principal Garu. Dr. Achita Devi Garu, who has rightly put the importance of this little technique that we'll be discussing subsequently, right? So I'm really happy the way that she has succinctly put the entire presentation in a couple of sentences. So emphasizing the importance of this technique is really heartwarming. Yeah, I thanks once again for that thing, Dr. Achita Devi Garu. I guess if she's listening to this, anyways. Uh, so I know that besides every program, there are many people who are in front and behind the screen. So let me thank all the conveners, coordinators, teaching staff, non-teaching staff, and of course my dear students and colleagues who are part of this conference. And I am sure you must have put in a lot of efforts to get this show running. So I'll try my best to do the job that I am entitled with. Okay, so if at all you find any problem with the way that with the language that I speak, in terms of the speed or in terms of the the language per se, please let me be aware of this so that I can change. And before I go into my lecture formally, I would like to thank one and all. I'm sorry if I missed anybody in congratulating or in thanking them. Please accept my personal namaskara for all of you for making this national webinar possible. Yeah, so with your permission, the coordinator, can I start my lecture?
Yes, sir.
Yeah, I'm just going to my screen sharing pattern and then I'll start it. Okay, so just give me a couple of minutes. Yeah. Well, for all of you who are new to this place, or I'm sure most of you might not know, we are 160 kilometers far from Guwahati, the capital city of Assam. And we had a very good rain yesterday. So the internet is a little flickering. So I hope it will cooperate and we'll get through this presentation at the quickest possible time. Okay. So yeah, I'm just trying to share the presentation. Just give me a couple of minutes. Yeah. So is it visible for all of you?
Yes, sir.
Yes, sir.
Yeah, I'm trying to magnify it and I just don't want to blow it out. Do you want me to blow it out? Yeah, I guess this is fine now.
Yes, sir.
So with your permission, I think let me get on to the show. So well, we here we are to discuss about a technique called as polymerase chain reaction. So I have selected the title as polymerase chain reaction, principle, types and applications, considering your requirement also for your graduate program. So I learned that PCR has been a part of this topic, has been a part of your regular BSc syllabus and also probably your master's syllabus. So I had made it more informative so that it will be good for you, and I'm sure you are going for your exams pretty soon. So this will be helpful for you. So let me at the upfront accept that I am no discoverer of PCR, nor I am just an application user of PCR. So I'm sure my colleagues here, if at all I fumble around, with the choice of words or with some technical help, you will be there to help me. And I'm sure you students can always get back to your teachers or my email ID whenever you need me for some clarifications on PCR for any other matter, can revert back to my email, right? So with this little introduction, let me get ahead. Let me go ahead and actually try to help you understand what a polymerase chain reaction is and what are the different types of polymerase reactions and what are the types of it. Before we actually study the type of the polymerase chain reaction, one needs to understand what is conservative DNA replication, what is semi-conservative DNA replication, and what is dispersive replication. So you may ask me that, sir, why should we know the types of DNA replication? Then my simplest answer would be that polymerase chain reaction is a technique, a biochemical tool where we try to amplify multiple copies from a single copy. And we use the principles of a typical DNA replication. So we try to simulate the in vitro conditions which usually happen in vivo, and therefore it becomes more necessary for us to understand the types of DNA replication. Now there are three different models of DNA replication which have been proposed till date. Now the first one being the conservative model. So what is a conservative model? We will discuss subsequently. Secondly, there is a semi-conservative model, and the third one is a dispersive model. So now let us see what is a conservative model. Yeah. So what does this strand or what does this conservative model of DNA replication say? The model or this conservative model DNA says that there is one template or one strand, one double helix DNA is present. This double helix DNA actually opens up and you have one parent strand and one newly synthesized strand, which means effectively you have 50% of new DNA and 50% of the previous strand. So if I were to further simplify, I would say that you will have one parent DNA molecule, which is double stranded in nature. You will have a new total double strand DNA molecule, which is synthesized newly. So this is what is called the conservative model. Now, why it was called probably conservative model is because you have the total one strand of DNA, the parent DNA molecule live with you and active. So this is what is called as the conservative model. Now, let us look at the So what do we get in this technique? It's pretty simple. So just like the word dispersive means scatter, the actual DNA or the target DNA or the template DNA which has to undergo replication would break, and then you would have a new strands of DNA being synthesized at that particular point to make it new, to make it complete. So if you look into this, you will have both the strands of DNA which will have a hybrid of or which are comprises of hybrid of old and new strands. Okay, so now let us look at the most accepted model, and this accepted model is what is known as the the semi-conservative model. So basically what they did was, the first was a conservative model where there is a parent double strand DNA and you have a newly synthesized DNA double strand. So you have one parent strand and one newly synthesized double strand DNA. Second one, the dispersive model, where the DNA is scattered and the rest of the DNA part where the scattering has been occurred, there the replication takes place and the DNA is totally repaired. So what happens there? In that process, you will have part of a DNA which is newly synthesized and part of a DNA which is the older one or the template DNA. Now, subsequently, the third method came into picture, which was more accepted because of the further Michelson-Stahl experiment. And that method was called as the semi-conservative method. So what is semi-conservative method of DNA? The semi-conservative method of DNA says that both the strands of the DNA are separated. Once both the strands of the DNA are separated, then this single strand of DNA would be serving as a template, and then all the DNA replication mechanism kicks in and you will have two new strands of DNA. So what happens? You will have one molecule of DNA with an old DNA and the other strand of the DNA which is newly synthesized. So effectively 50% of each double strand DNA will be new and 50 will be the older DNA. So this was the most accepted model, because and the support to this model actually came from the Michelson-Stahl experiment. So what did Michelson-Stahl do? They gave a radio nitrogen. See, we usually have nitrogen 14, but what these people have done is they used N15, nitrogen 15, and once this N15 was given for an actively dividing cell, the cells would take up these nitrogen bases which have N15, and after the cells undergo multiple cycles of replication, the cell pellet was taken and subjected to density gradient electrophoresis. Then they found out N14 band, N15 band, and N14 and 15 band, suggesting that there is an N15, there is a DNA which is the parent strand, N14 which is the parent strand, N15 which is the newly synthesized strand, and you have N14 and 15 both as a hybrid. So this experiment of Michelson-Stahl actually laid the foundation or gave a scientific conclusion with an experimental data about the semi-conservative mechanism of DNA replication. So now looking at after looking into this, let us go into the standard definition of what a PCR is. See, the definition of PCR is very varied because it's like defining what is a computer. See, in the beginning, it's like a compute. What was computer definition? The definition of a computer was an instrument or a machine used to calculate. Now it does many things beyond just calculation. So similarly, PCR is just I'm just showing you the statement here, which was given probably long back. But now there are many modifications of PCR, that we may have to redefine each and the statement for each and every of this PCR. So as of now, let me introduce to you what do you mean by PCR. Now, PCR is a biochemical technology wherein a small amount of template DNA or a small amount of DNA, which we call target DNA, it could be very small, it could be a huge fragment, and sometimes an entire chromosome, is used to amplify in a significant amount of quantity, which means you are taking the DNA and you are amplifying it to probably more than 1,000,000 times. Why do you want to amplify it? Because you want to study that amplified DNA in a systematic way for whatever application it may be. It could be for identifying whether a pathogen is present or not. It could be trying to study the mutation, if at all there is a mutation existing in the parent DNA or not. It could be for multiple other reasons. So again, the question comes, why do we do a PCR? This is a very fundamental question which many students ask me. See, many times we have a problem with the sample. In biology, getting the sample or availability of sample is very low. I think your teachers, when you are doing experiments also, they may bring a plant. Most of you guys are from plant. You know that there are some plants which are very rare. There are some plants which flower only very rare. So if your teacher struggles to get that flower after traveling, let's say 200 kilometers, and if you don't get and you can't afford to lose that sample, right? So many times the availability of the sample is a very important aspect. So if the availability or if the quantum of the sample that you have is very low, the only way, and more specifically the DNA is very low, and if you want to do your analysis on that DNA, it may not be sufficient. So PCR is a technique where you could modify or multiply, not modify or multiply this small quantum of sample to a huge quantum which can be used for your analysis. So that is one. Second is, that getting the sample may be a big jog and sometimes risky. Like I told you, if your teacher wants to actually study the evolution of a particular plant, and if she has gone to a very far mountain and got a sample, and she would hardly be in a position to lose that kind of sample. If she could use a PCR technique, she could amplify the DNA of that plant and to a whatever required level, and then she can do all the analysis that she that she wishes to do. So these are two major reasons why we do a PCR. Yeah. Uh, can somebody mute? Is somebody asking anything?
No, so you can continue.
Yeah, I can hear some background. So I thought if somebody wants to ask something, they can play, because I have magnified my this thing, so I don't see anybody raising hand or something like that. So if there are any questions, please forgive me. I will answer to them at the end of this lecture. Right?
Yes,
Yeah.
sir.
So I know this is going to be a monologue, but then there is no alternative to this, right? So, well, then the immediate question comes, who discovered PCR? See, it was Carrie Bank Mullis who discovered the PCR. And you will be surprised to know there is a short story associated to this. He was an American biochemist, an author, and a teacher. Now, in recognition, it is said that Carrie Bank Mullis was a scientist who he discovered the idea of a PCR when he was going on a ride with his female friend, or I would say a wife probably. right? See, who on this planet would actually think of science, when he is traveling with his girlfriend or with his wife? I think only a scientist. So one should appreciate that it is only the scientist who can always do multitasking. He can have science parallel running in his brain with his domestic and other activities. So we should really appreciate for Carrie Millis. I'm sure he may have spoiled his evening on that day when he was traveling with his the better half. But unfortunately or fortunately, today we have PCR because of one that little thought. And in recognition of his discovery for PCR, he shared the Nobel Prize in Chemistry in the year 1993 with Michael Smith. And in the same year, he was even awarded with a Japan Prize, right? So this is the one who discovered the PCR. And Dr. Carrie Bank Mullis was the biochemist. Now let us look into Carrie Bank. Let us further look into what he has done. See, in 1983, Mullis was working for Cetus Corporation as a chemist. And Mullis succeeded in demonstrating the PCR on 16th of December 1983. Now in 1986, remember, Carrie Mullis performed PCR without tag DNA polymerase. Now subsequently in 1986, when Thermos Aquaticus, when tag DNA polymerase was identified, then only then from the Thermophilus Aquaticus, only then the tag DNA polymerase was used. So let me tell you that tag DNA polymerase was not used when actually the PCR technique was discovered. Now it was Randall Psyche who generated the needed data and authored the first paper to include utilization of this technique, while Mullis was still working on a paper that would describe PCR itself. So this is what the whole contribution of Carrie Mullis was. Now let us see what are the requirements of a PCR. What are the requirements of a PCR? One is a thermal cycler. So one is a thermal cycler, and then you also need a micropipette, plasticware, glassware, and a sterile work area, a template DNA, a tag DNA polymerase enzyme, DNTPs, buffer, primer, and sterile water. Admin, is the admin active? Admin?
Yes, sir. Yes, sir.
Can you please block Kirti?
Yes,
I
sir.
think her mother is doing something for her.
No.
Yeah. Well, I was getting a little distracted. Anyways, so thermal cycler, you need a thermal cycler machine, you need micropipette, you need plasticware and a sterile area. The most important thing that you would need was a template DNA, and you would also need a tag DNA polymerase, of course. Besides which, you need DNTPs, buffers, and divalent ions. You need a forward and a reverse primer, and sometimes you need even sterile water or deionized water to make up the volume if necessary. So these are some of the requirements for performing a PCR, right? So let us look into this organism, this little organism from which we had isolated the tag DNA polymerase. See, the organism from which tag DNA polymerase was isolated is known as Thermos Aquaticus. So in 1969, Thomas D. Brooke and Hudson Freeze of Indiana University reported a new species of thermophilic bacteria, which they named as Thermos Aquaticus. Now, this bacterium was first discovered in the lower Geyser Basin of Yellowstone National Park. I'm sure you all know where Yellowstone National Park is. It is in United States of America. Now near the major geyser, Great Fountain Geyser and White Dome Geyser. Now, Thermos Aquaticus is a species of bacteria that can tolerate high temperature, meaning it is a thermophile that belong to Deinococcus thermus group. This bacteria is a chemotrope and can survive up to 70 degrees, but can survive at temperatures between 50 and 80 degrees. Now, it is the source. It is from this organism that the tag DNA polymerase was isolated, right? So, What is the beauty? In 1976, probably they have isolated the tag DNA, this organism, that is Thermos Aquaticus. Many scientists or many students know Thermos Aquaticus organism only for identification of associated with tag DNA polymerase. But let me tell you, there are many other important biological enzymes which have been isolated or extracted from Thermos Aquaticus. And some of them include aldolases, RNA polymerases, TAC1 restriction enzyme, and DNA ligases. Right? Now, let us now look at the properties of tag DNA polymerase. See, it is this tag DNA polymerase which is actually responsible for the synthesis of the new template DNA. So, what are the properties of tag DNA polymerase? See, tag DNA polymerase can replicate 100 bases of base pair strands of DNA in less than 26 at 72 degrees, which means in 10 seconds, 1 kb of DNA can simply be amplified at 72 degrees. You can understand the speed at which it does this job. Now, this enzyme is highly thermostable, meaning the half-life of this enzyme is around 2 hours at 92.5 degrees. Now, at 75 to 80 degrees, tag DNA reaches is optimal polymerization rate and about 150 nucleotides per second. Now, at temperatures above 90 degrees, tag demonstrates very little or no activity. Remember, here is an enzyme which does not actually denature or remains intact even above 90 degrees. Now, this is what a typical thermostable enzyme is all about. Now, generally, potassium ions and magnesium ions promote the activity of tag DNA polymerase in optimal concentrations. However, if you increase the concentration of these ions, they inhibit the tag DNA activity. It is like this. You may like a sweet, 1 or 2 pieces of sweets, but if I give you 10 or 20 pieces of sweets, you may not actually like them. So similarly, tag DNA polymerase has good activity or increased activity with certain concentration of magnesium chloride and potassium chloride. However, with increasing concentrations of these, they may actually inhibit the activity. Now, EDTA also binds to the tag DNA in the absence of these metal ions. Now, the biggest challenge or the biggest limitation what people actually encounter when they are working with tag DNA polymerase is tag DNA polymerase does not have 3' to 5' exonuclease activity. So effectively, therefore, tag DNA polymerase has low replication or relatively low replication fidelity or no proofreading activity, if you would ask me. Now the error rate is that 1 in 9,000. In general tag that you are using, it is about 1 in 9,000 nucleotides. So tag DNA polymerase is a very good enzyme per se, highly thermostable, but the problem is that it does not have a proofreading activity. So if it is not having a proofreading activity, then in the samples that you are amplifying, you may have issues. You may have some unwanted mutations coming in when you are replicating. So this is one of the biggest challenge with respect to tag DNA polymerase. The other problem with tag DNA polymerase is, you can call it as a problem or you can call it as an advantage, is that when you amplify a product with a tag DNA polymerase, you get overhang of A. So this A in a PCR product would probably help you in doing a TA cloning by a sticky end cloning method, one of the method. However, if you want to do a blunt cloning method, amplifying the PCR product using a tag DNA polymerase may be a problem, right? Now, we know that tag DNA polymerase is a wonderful enzyme, but it has its own limitations. And like I told you, there is a problem of proofreading activity. So one needs to look for substitutes. When scientific community started looking at substitutes, they found out another thermostable enzyme known as PFU DNA polymerase. Now, PFU DNA polymerase has probably all the properties required for amplifying or for replicating the DNA like tag DNA polymerase, but it has its own advantages. That is, PFU DNA polymerase has a proofreading activity. But the problem with PFU DNA polymerase are these. PFU DNA polymerase is a slightly lazy enzyme compared to tag DNA polymerase, right? You can see PFU DNA polymerase in PCR reactions results in a blunt end PCR product. So you cannot do a TA cloning, but you have to only do a blunt end cloning. You cannot do a TA cloning, or a sticky end cloning is not possible. PFU DNA polymerase can also be used in conjunction with tag DNA polymerase, meaning you can take some PFU DNA polymerase and some tag DNA polymerase, and then you can have a pool of the DNA polymerase which will have the fidelity, and also you can achieve the speed of a tag DNA polymerase. So these are the limitations and advantages of tag DNA polymerase and PFU DNA polymerase. So why I am stressing on tag or PFU DNA polymerase? Because in many of the interviews where you attend for your masters or for your competitive exams, people ask, so one of the limitations of tag DNA polymerase, or few of the limitations of tag DNA polymerase, or PFU DNA polymerase. So kindly look into this, and this will be very advantageous for you. Now, how does a typical PCR work? So here I am going to tell you how a PCR machine works. While the biochemistry of the PCR is different, here I am speaking about the instrument per se. How does a PCR instrument work? Now, a thermal cycler is a laboratory device used to create the polymerase chain reaction process, which in turn amplifies DNA segments. A Peltier thermal cycle is a simple thermal cycler which employs Peltier heating and cooling elements in order to control the temperature. The addition of Peltier heating or cooling elements allows for greater temperature precision and makes a thermal cycler possible. I was requested by your teacher to actually show you a live demonstration of a PCR, but I'm really sorry. Most of the students in the university are out because of COVID-19, and therefore I am not able to show you the live demonstration of a PCR technique. Otherwise, I would have definitely requested one of my students and shown you a video of that. But however, there are many YouTube videos which are available, which you can browse, and they are mostly free. So you can simply browse them and get the required information of how to do a PCR. Right now, what are the different steps involved in a PCR machine? See, there is the primary step called the initialization step, and then the second is the denaturation step. The third is the annealing step, extension or elongation step, the final elongation step, and the termination step. So these are the entire steps that the whole PCR works. And there is a repetition of cycles from step number 4 to step number 2 to around 25 to 30 times. So I will tell you what happens in each step in the subsequent slide. So these are the typical PCR reaction, or the steps that are there for a typical PCR reaction. The first is initiation, or sometimes called denaturation step. So what is a denaturation step? You are heating the DNA to around 90 to 95 degrees Celsius. So why 90 to 95 degrees Celsius? You want the entire DNA which you want to amplify being separated into 2 strands by 30 to 60 seconds. Now what you would do is you would take a forward and a reverse primer, and then you would add them to the PCR reaction. Since they are already present, this annealing temperature, you would bring down the temperature of the entire reaction mixture between 50 and 65, so that the primer goes and binds to your target DNA. So once it binds to the target, you give not more than 60 seconds to achieve this kind of objective. So once this is done, you give an extension between 70 to 72 degrees for 30 seconds. Why? We know that the tag DNA polymerase has maximum activity at 72 degrees. Some people give even for 1 minute also here. And this step between 2 and 4 or 1 and 4 is repeated for multiple times. Usually they are repeating between 25 to 35 cycles. Now why 25 to 35 cycles? Is for the simple reason that for each cycle of PCR, there is an amplification which gives in 2 to the power n, which means if the PCR undergoes one cycle reaction, 2 to the power of 1 is 2. If it undergoes 2 cycles, it is 2 to the power of 2, it is 4. If it is 2 to the power of 3, you would get 8 amplicons. Similarly, by the time it reaches the 25th cycle, which is 2 to the power 25, then you would have already got 1 million cycles. So people generally put between 25 to 35 number of cycles. Now the final elongation is between 72 degree at 72 degrees between 3 or 5 minutes or 10 minutes is for the simple reason that PCR, the enzyme may not have amplified some part of the fragment because of losing it some of the activity. So we are allowing the enzyme to complete the process of amplification by giving this final elongation step. So we are repeating this step between step number 1 and actually 5, so that this happens every other time. And finally, we are actually terminating that step or terminating the reaction by bringing down the temperature to 4 degrees. So this is how a typical PCR reaction runs. Now everybody has got does minor variations either with the annealing temperature or with the elongation temperature, sometimes or time of elongation, or with the number of cycles, or with minor variations with respect to denaturation. But this is a typical PCR reaction which I wanted to show to you. Now, subsequently, I wish to show you a video how PCR actually is done. Please look into this. The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a primary state of DNA polymerase called tag polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the tag polymerase. So you can see that the DNA is the template DNA and you have a target gene of interest to be amplified. So you need an enzyme, you need DNTP, and you need other bivalent ions to get the amplification done. So you see that the forward and the reverse pairs of primers actually go and bind once the DNA is denatured. And then the tag polymerase actually synthesizes the complementary strand of DNA using the fiber as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. So this concept of heating and cooling so that the DNA is denatured, the primers would come and bind and then would amplify the segments of DNA is what happens. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycle, an instrument that automatically raises and lowers the temperature at a low. So while this video says that overnight, but these days you have instruments which can do this amplification in not more than 2.5 hours, right? So depending upon the kind of instrument or the type of company brand of instrument that you buy, You can get this PCR done. You can get 1,000,000 copies of DNA from a single strand of DNA within a span of 2.5 hours if all things are fine with you, if all the reagents and if all other setup is okay with you, right? So this is an animation of PCR where I would like to show you how PCR is actually how PCR helps in amplification, or what is the principle behind PCR. Now there are many applications of PCR, and here I have listed few, and they include uh, you know uh, and they include selective gene isolation, which means you can selectively isolate the gene, you can amplify it, you can do a gene cloning, and subsequently that cloned you can clone that particular amplified gene into rDNA, into another vector of your choice. You can use this PCR for medical diagnosis, which means you can actually find out whether these see, if you are designing a primer targeting a particular organism. Let us say I want to test for certain bacteria in you. So I will take your blood sample. I will design the primers for these, these organisms, and then I would isolate the DNA from your blood sample. I would put the PCR reaction using the primers that are specific for the bacteria, and I would do a PCR. Within 2.5 hours, I get the amplified fragment. I run it on an agarose gel, and then I can see if I get the desired band. Don't you think that organism is present in your body? Yes. And if there is no amplification, I can simply say that that bacterial organism is not present in your body. It can be a bacteria, it can be a fungi, it can be whatever organism that you are planning to look for. The only challenge is designing and obtaining the right fragments of DNA and getting the good and clean sample that you want to test. Remember, in forensic medicine, you must have heard that there are few droplets of blood which actually fall off, at the crime site. So these small volumes of blood are used, taken up, and then the DNA is extracted, and then things are being used to find out who is the culprit behind this. Right? So, and many times in conservation biology also, like I told you, if you want to protect a particular organism, whether it is a plant or animal or whatever, you can do that with a PCR technique. So there are multiple applications of this technique, and there are also few applications in medical diagnosis, just like I told you. In fact, not only the presence or absence of DNA, you can also sometimes quantitate the bacterial load. What do you mean by bacterial or viral load? You can find out the amount of DNA, that is, you can probably estimate the amount of DNA or the DNA, and then indirectly quantitate the number of bacterial colonies or number of viral organisms that are present in a given volume of blood sample. So this makes the PCR a very important tool in medical diagnosis. So you have umpty number of applications of PCR in a conventional biological systems and also in medical diagnosis systems. Now, but there are very few limitations also associated with this technique. Now, what are the limitations of this technique? Is that in order to amplify, you need a target DNA. So without target DNA and the designed primers, you can never go ahead and do a PCR. Secondly, DNA polymerase or tag DNA polymerase is prone to error, which means it can create mutations. So if you want to take this PCR product, which is amplified by tag DNA polymerase, and if you want to do a cloning, then you are at a problem. So you may have to use a modified DNA polymerase or a PFU DNA polymerase to get your job done. Now, the problem with this technique is it is highly sensitive. So even if there is the smallest contamination of an unwanted DNA, and if your primers are binding to that, you may get an ambiguous or misleading result. See, nowadays we are all seeing that, there is one patient who is corona positive, has the symptoms, but he is tested negative. And there are people who are tested positive, but are actually negative. And one of the reasons could be that there is a contamination, or there could be some contamination at some level, either while taking the sample or either while performing the experiment or wherever. I don't say that all of them are because of errors. Sometimes it could be, or it could be one of the reasons. Many times there are samples also which can actually, sometimes the samples contain organic molecules which inhibit PCR reaction. See, you go to a pond, you collect water sample, you want to isolate a DNA from a bacteria in a pond, but that besides that organism, there are certain other salts or other acids which are present in the pond water, making it unfavorable for the DNA to actually get amplified by PCR technique. So there are many, the advantages for PCR technique. However, there are few limitations as well. Right now, if you were to study the different types of PCR machines, I have shown around 10 different types of PCRs. However, not all of them are used in great demand, or not all of them are in great demand, except only very few of them are actually routinely practiced. See, what is multiplex PCR? I, I told you, when we studied at the video, we found out that a typical PCR only uses one pair of primers. But remember, in a multiplex PCR, we actually use more than one pair of primers. What is a hot start PCR? I will discuss multiplex PCR in a in subsequent slide in a much more vivid way. So let me tell you, multiplex PCR, hot start PCR, long range PCR, loop mediated isothermal amplification, reverse transcript PCR, real time PCR, high fidelity PCR, fast PCR, GC rich PCR, and arbitrary primed PCR. So many types of them, right? So let me go first with multiplex PCR. What is multiplex PCR? Multiplex PCR is a PCR where you are actually trying to use different templates or different primers for a single target, right? And you're amplifying them to get your desired amplifications in a single reaction. It means you are taking a big chunk of DNA, you are isolating or you are amplifying fragments of DNA of your choice. So unlike a conventional PCR where you are using a single primer, here you are using multiple primers. There are certain prerequisites, that means certain conditions which are required if you would actually want to do a multiplex PCR. What are they? It is that you should actually use primers which are non-interfering. You should use primers which have common association or melting temperatures. Otherwise, if one primer is having an annealing temperature of 56 and another primer is having an annealing temperature of 60, then you will have a problem. Yeah, and each amplification proceeds independently, and then you get a desired fragment. So basically you should also take into account that these primers should not bind to each other. If they bind to each other, then there is no process of by which you can actually make these primers and go and bind to the DNA at specific points and get your desired fragments. So these are the different requirements or requisites or prerequisites for a multiplex PCR. However, there are many applications. The first infectious disease application of PCR was detection of HPV, that is human papilloma virus, using degenerate primers. Gregoria and co-workers used degenerate primers containing deoxyinosine at variable base locations and showed that consensus primer could detect all HPV genotypes, which means in this slide, I have shown you few reports which says that by multiplex PCR, you are using them in clinical setup, right? So you are using multiplex PCR and finding out the presence or absence of a particular organism in an individual. So this is what is the application of PCR or modified PCR, or for that matter to be more specific, multiplex PCR. Now, I want you to show you a video of another type of PCR, which is known as lamp or loop mediated isothermal PCR. Now, the biggest challenge with PCRs is when you do a typical PCR, I told you, you need to buy an instrument. You need to have a clean, sterile environment. You need a lot of plasticware, glassware, and technical expertise. The biggest challenge is to procure an instrument. Now, if you want an instrument, it will cost you around 2 to 2.5 lakhs these days. Of course, you have a lower, cheaper version or less expensive versions of that. But even the most less expensive version will cost you 1 lakh rupee, I guess. So in order to actually, eliminate some of the processes of this, what people have discovered is a modified or a new technique which is known as LAMP. Please look at this video where I am trying to show you how LAMP works. So what happens in loop mediated isothermal amplification? I have used a video from NEB. So what actually happens in this kind of technique is that you are using at least 5 or 6 pairs of primers and you are amplifying the DNA, right? So the biggest advantages of this technique is that you don't need a PCR machine, since there is isothermal amplification, meaning that you are amplifying the product or amplifying or performing the PCR at a single temperature. So a simple water bath at a fixed temperature will do all what is required for you, right? So you need not invest in purchase of a big and expensive instruments like a PCR machine, right? So they have been extensively used in the current technologies. Now, like I have mentioned here, it is an isothermal approach to nucleic acid amplification using a single temperature and does not require a thermal cycling. Now, as it is not limited by doubling cycle amplification, LAMP generally produces more DNA than PCR in a more rapid incubation time. Now, you may not even need an agarose electrophoresis to do. Sometimes you can simply use a turbidometer, or with a naked eye, you can detect the presence of a target DNA. So this is very, very, very important and advantages of the LAMP technique. However, there are limitations. Like any technique, this technique also has few limitations, and the limitations include that it is less versatile than PCR. It is useful primarily as a diagnostic or detection technique, but not for cloning purposes. meaning you don't use this technique for cloning purposes. Now, multiplexing approach for LAMP are less developed, which means to design the primers for multiplex PCR, there are not many online free tools available. Similarly, the larger number of primers per target in LAMP increases the primer-primer interactions. So these are the few limitations of LAMP technique. Right. So now we have studied, one is a multiplex PCR, and second is a loop assisted loop associated PCR. Now I have a loop mediated isothermal amplification, or LAMP PCR. Now besides this, I told you we have something called as hot start. Or what is hot start? You are putting the PCR, you are starting the PCR only when the PCR machine reaches to 95 degrees, so that you don't want unnecessary binding of these little primers at the undesired positions. Long range PCRs are those PCRs where you are trying to amplify a long product of PCR, which means that you may be interested in amplifying as big as an entire chromosome. Right? High fidelity PCR is a PCR where you are actually trying to amplify a fragment which is totally foolproof. I mean, which is totally fiddle, which means it is a copy of what you are trying to amplify. You are using a good quality and an enzyme which has got a proofreading activity. Fast PCR is the PCR where you do it in a faster way to get your desired products and many things like that. So now let me also go to a little more other topic, is what is known as reverse transcription PCR or RT-PCR. Now, this is the PCR which is spoken day in and day out by many people, whether they know its science or whether they don't know science. It is because of this corona or COVID-19 situation, we all are exposed to these words. So let me explain you what is an RT-PCR. Now, RT-PCR is a laboratory technique which combines a reverse transcription of RNA to DNA and subsequent amplification by PCR. So there are two techniques in this. One is you are doing a PCR to amplify or to find out whether a particular organism is present or not, and also to quantify it. What do? This technique is usually employed when you are working with RNA samples. What does this technique do? It is primarily used to measure the amount of specific RNA and also find out the presence or absence of a particular RNA species. Now, how is this done? It is achieved by monitoring the amplification reaction using fluorescence, a technique called real-time PCR or qPCR. So first what you do, you would do a reverse transcript PCR. What is reverse transcript PCR? You are converting an RNA into a cDNA or a DNA. C is cDNA or from RNA to DNA, and then this DNA is used as a template, and then you actually do your PCR. So a combined RT-PCR and qPCR are routinely used for analysis of gene expression and quantification of viral RNA in research and clinical settings. So this is what people are doing day in and day out to find out whether you have corona positive or whether you are COVID-19 positive or not. Right? So it is not as simple as you think. And it is also not complex as you think. Okay, so what are the steps involved in it? The first step is you have an RNA template, and this RNA template is that of the virus that you are actually thinking of. You are using an oligo-DT primer and you are amplifying the entire region using a reverse transcriptase or an enzyme which is an RNA-dependent polymerase. Till now, tag DNA polymerase was DNA dependent polymerase. Here you are seeing an RNA dependent polymerase. So you are using RNA template, you are adding a oligo DT primer, and then you are amplifying the entire region. So once you get this entire region with this RT, then you are actually having a RNA DNA hybrid. What is this? This is an RNA template on which you are actually generating a DNA molecule. So you are getting a RNA DNA hybrid. So this cDNA is what is then subsequently double-stranded or made double, and a complementary DNA is synthesized, and then you do the amplification of the gene that you are interested in. So this is what is known as reverse transcriptase PCR or RT-PCR. Now, what are the challenges? Like advantages we have already seen. It is used in clinical and laboratory settings. It is used in finding out whether you are a disease positive or a disease negative. You can quantify the RNA. You can also look for the presence. Now, what are the limitations of this technique? That is, the exponential growth of the reverse transcript complementary DNA during multiple cycles of PCR produces inaccurate endpoint quantification due to difficulty in maintaining linearity. Even the slightest DNA contamination can lead to undesirable result. So this is what you are all now seeing. You are saying, I am finding that somebody is RT negative, RT-PCR negative, but still he has disease symptoms. It is because of this. A slightest DNA contamination at any point, a slightest contamination at any sample collection center might give you these kind of results. Now, planning and designing of quantification studies can be technically challenging due to existence of numerous sources of variations, include template concentration, amplification, and multiple challenges. And it is not an easy one. Had to RT-PCR machine is also pretty expensive. One needs to have really an expertise when you are working with RT-PCR. So these are some of the advantages or types of DNA applications or PCR applications and types of PCRs that I have tried to highlight. So with this, I would like to acknowledge my department, that is Department of Molecular Biology and Biotechnology, Tezpur University, for all the support that they have given me. For all that I am here, and I am a faculty of this university, all the administrative staff and others. And I would quite honestly and humbly accept that all the videos that I have shown you are not made by me. All the content that I have used is from internet. So I would really, from the core of my heart, acknowledge all the original content creators whose images, videos, texts that have been used in this presentation to make it effective, and so that you students can learn it in a better way. Right? So I strongly believe in this single statement where there is no substitute for hard work. One has to really do hard work, and only then you can succeed in life. And here is my favorite Sachin Tendulkar, and my more favorite is Vishwanathan Anand, who is a grand chess master, grandmaster from India. So I really appreciate both of them for their untiring efforts in and making this country proud. So I'm sure there will be a day where one of your students' picture will be here, and I'll be, as a teacher, sharing to other students this. I hope you come on this slide someday. And this is what really inspires me. A statement by Swami Vivekananda, or popularized by Swami Vivekananda: arise, awake, and stop not till the goal is reached. So with this thing, I would like to thank you for your kind attention and all that you have been listening to. And I would request all the teachers who are there to help the students if they have any doubts, or you also can actually write to me, and I'll be happy to solve the doubts if it in any. And I am sure there is no alternative to direct teaching, but given the present situation where we are in, I think there is no alternative but to do these kind of seminars in the best interest of the students and the faculty. So I would once again thank all the organizers, the principal madam, the coordinators, the co-conveners, conveners, teaching staff, non-teaching staff, and all the participants who have made this presentation, who had made this webinar probably a successful one. So I am exiting my screen sharing here, and I am willing to take the questions if at all, if there are any. Yeah, please.
Thank you so much, sir. It was very informative to us, and thank you for giving your valuable time to us.
Thank you.
Now, I invite Kirti Ma'am, Assistant Professor, Department of Botany and Food and Nutrition, to take over the questionnaire. Over to you, Kirti Ma'am.
Thank you, Renu. Sir, there is a question from Tirupati sir in the chat box, sir. How much, to what extent we can depend on RT-PCR for detection of COVID-19? This is the first question, sir. And the next question is why we are now along with this technique called CT scan.
Well, how much depends upon RT-PCR for detection of COVID? See, Tirupati Garu, thank you for this question. It's very, very, intelligent question, I would say, and thanks for raising this pertinent question. See, basically, RT-PCR is highly sensitive. The sensitivity of this RT-PCR actually depends upon the primers that you are actually using. For that matter, not only for COVID-19, any organism that you want to test, if your primers are unique and are specific to your target DNA of the organism, then it is highly unlikely that you would get a false negative result. Unless and until there are some other technical issues like sample contamination or any other, non-specific binding, etc. But if your primers are fine, and if they are specific to your target DNA, then definitely no chance of going any, there is no turning back. I'm sure this is one of the best technique that you can rely on, right? Yeah, of course, there are challenges for RT-PCRs per se, but I'm sure this is what it is. So why we are now along with this technique, chest scanning is, see, always a diagnostic text. I am not the right person to answer this question because I am not a clinician, but with my very little understanding, a diagnostic RT-PCR is a diagnostic technique. So probably the clinicians also want to ascertain the status, the clinical status of a patient by looking at the chest, by CT scanning the chest also. So that will be a double advantage for the clinician to come to a logical conclusion. And this is what I presumably believe. But otherwise, RT-PCR is a very reliable technique. It can give you the correct results provided your primers and sample is fine, right? I hope I have answered that question.
So we have another question from Ananya sir.
Yeah.
Can all the infections can be diagnosed through a PCR technique?
Well, all infections, I don't know. See, if at all you are looking for a microorganism, and if you know the sequence of the microorganism and can design the primers for that, definitely yes. See, let us say you have found out a bacteria and you know the sequence of this bacteria, only then you can design the primers. And when you can design the primers, and if they are specific and good and optimized, then obviously you can go for PCR technique. No question about it. I think this is the answer that I can give to Ananya. Yeah.
Thank you, sir. There is another question. What will be exclusive properties of primers for both forward and reverse primers?
Well, exclusive properties, it's not called exclusive properties. They are called what are the rules for designing primers. I have not specified here the rules of designing primers, but there are certain rules. There is a book called PCR Protocols. You can read that book. They will tell you what are the different rules. For an instance, I will tell you that primers should not be too short or too long. Generally, primers actually are between 6 and 20 nucleotides. You can have a longer primer, but below 6, if you use, you may have non-specific amplification. And if you use beyond 20, again, you will have non-specific amplification. The forward and reverse primers melting temperature, the difference between the forward and the reverse primers melting temperature should not be beyond 5 degrees. This is another rule. Thirdly, that forward and reverse primers should not have high percent of GCs. Fourthly, you should not have primers which actually form hairpin loops. Fifth, you do not have primers which are self-aligning in nature. So these are some of the properties of primers actually. Yeah. Yes. What is the difference between rapid test and RT-PCR? Madam, rapid test is an antibody-based test, I presume, and that is what is being done, right? Of course, there are different proteins for which antibodies are raised, and then you would do a rapid identification test, probably because of that. But RT-PCR is a DNA-based test or a PCR-based test where you actually convert the cDNA to the RNA to cDNA and then to amplify using a conventional PCR. I guess I have answered your question, Madam Kavita Garu.
Yes, sir. Yes, sir. But actually, sir, which is most prominently accepted, sir?
See,
Is it rapid test or RT-PCR?
antibody based tests are always little tricky, right? PCR is a very, very, very sensitive tool, and RT-PCR is much more sensitive. And that is why for detecting COVID-19, RT-PCR is the gold standard, which means you have to rely on RT-PCR data.
Thank you, sir.
RAT may be little dicey.
Okay. Thank you, sir.
Yeah, please. Yeah. Anyone, please? I have actually, there are a lot of my students who have probably wanted to join here, but then because of the limitation of number of students, I think they are watching it live on YouTube, which you are this thing. Yeah, I
Kirti,
think.
even you can take questionnaire in the YouTube also, Kirti, if there are any questions.
Yeah,
Yes, ma'am, I'll see.
which is the most effective technique deciding the COVID, Anita ji. So I think RT-PCR is the gold standard currently followed by most of the government agencies. So our RAT is not. It is a preliminary screening technique, is what they say, and COVID-19 ke liye they are using only RT-PCR as a gold standard. So I think this is what is the current one. Yeah, I hope I have not exceeded the given time. I just stuck to the time 1 hour time limit. So probably from 11:30 to 12:30 I was there, and if there are still few questions, I can take it.
Yes, sir. Thank you, sir. Thank you for answering our questions patiently.
So
Over to you, Renu.
before I actually wind up, I would like all the students to get back to your teachers, and your teachers are very, you are lucky to and proud to be. You should take the pride of being an RBVRR students, and you should be lucky to have such intellectual teachers with you. So please, you can ask them. They are very helpful, as I can see from the interaction with them. So, and I am always there with you students to help you and answer whatever I can. But yeah, in these COVID times, actually, I think you should take help from your teachers more than what you actually do. Yeah, madam, please, if there is anything else.
I request Ushasri, Assistant Professor, Department of Botany and Food and Nutrition, to purport vote of thanks.
Good afternoon, one and all. I, A. Ushasri, Assistant Professor of RBVRR Women's College of Botany and Food and Nutrition Department. It's my privilege to have been asked to propose a vote of thanks for this webinar. First and foremost, I would like to thank our today's guest speaker, Dr. Surya Prakash G. Ponnam sir, for accepting our invitation and sparing his precious time for exchanging his valuable knowledge on polymerase chain reaction principle, types, and application. We are grateful to our Professor Muttam Reddy sir, Secretary HMVS, Professor Sudarshan Reddy sir, Secretary cum Correspondent of RBVRR Women's College, Dr. J. Achyuta Devi ma'am, Principal of the college, Dr. Kavita ma'am, Vice Principal of the college, for their guidance, support, and encouragement. My heartfelt thanks to our HOD, Dr. A. Rajni ma'am, for her interest, vision, and commitment in organizing this webinar. I also extend my thanks to all the faculty members and our beloved students for their cooperation and attention. Thank you all.
Thank you very much, Ushasri Garu.
Thank you, sir. Thank you. Excellent session, sir.
Thank you. I hope it was informative.
Yes.
Thank you, sir. We are expecting this webinar. Thank you so much, sir.
Thank you very much, Rajni Garu. So I guess if it is done, can I leave, please?